Hello and welcome to this video on the interesting relationship between the FDA and biohackers. For those not familiar with these parties, the FDA is the Medical, Pharmaceutical and Research Administrator of the United States of America. They govern, enforce and monitor adherence to legislation. On the other hand are biohackers. These range from university-trained, highly regarded and well-known researchers to science enthusiasts. The problem is largely the latter group. These are often untrained, uninformed, and working out of their backyard sheds to genetically transform organisms. This is a niche area where the means and resources have been restricted, were prohibitively expensive or non-existent for a very long period of time. At least this was true until the mid-2000s. At that point, reagents became readily available, and later, machinery has become available. In about 2013, the open-source PCR machine became readily available for a modest cost. The combination of low cost, availability, and the ape's inquisitive mind led people to begin playing with these machines. Sometimes harmless experiments with green fluorescent proteins, and other times something less innocuous. As a result, at least 2 companies offer DIY gene editing kits. This is also where the FDA is concerned. To quote an article by Fast Company, as he offered samples in small baggies to the crowd, Zayner explained that it took him about 5 minutes to make the DNA that he brought to the presentation. The vial held Cas9, an enzyme that snips DNA at a particular location targeted by the guide RNA in the gene editing system known as CRISPR. In this case, it was designed to knock out the myostatin gene, which produces a hormone that limits muscle growth. This lets muscle atrophy. In a study in China, dogs with the edited gene had double the muscle mass of normal dogs. If anyone in the audience wanted to try it, they could take a vial home and inject it later. Even rubbing it onto the skin, Zayner said, would have some effect on cells, albeit limited. End quote. This kind of availability and distribution is a source of concern. It concerns authorities like the FDA, not only because humans can modify themselves, but also animals. GMO animals are heavily regulated across the world. Futurism summarized this as follows. To begin the quote, many of these DIY scientists are focused on improving animal species for better breeding, but some skeptics are afraid some well-intentioned scientist, somewhere, might accidentally homebrew a deadly pathogen or open some other sort of Pandora's box of genetic devastation. End quote. The case of Zayner mentioned at the start is an example of this occurring in humans, where the myostatin gene, if it is deactivated, may cause considerable pathogenic activity without being able to treat it. You may wonder why this has happened, and the reason is relatively simple. Many of the laws regulating genetically modified organisms, animals, and gene editing can only be applied to institutions, labs, and companies. The home DIY biohacker was not and is yet to be fully covered by these laws. Regulation by the FDA would allow them to keep working on their makeshift labs and in their makeshift labs, but they would be required to have their genetically modified products vetted by the FDA. This is the same process that is required for agencies that want to bring in new drugs. As it stands now, the equipment and reagents are available at a modest price, and they are often purchased by schools and biohackers for relatively harmless activities. But there is a concern, and a growing one at that their activities may prove to be less harmless over time. This is all occurring at the same time as the FDA is approving the first human genetic modifications for treatment of diseases, specifically acute lymphoblastic leukemia, which has shown significant effect and great benefit to those who are being treated with it. You will find a number of links below to look at for the reviews of this particular activity, the FDA's governing documents, and the machinery that was discussed, and the suppliers that provide the reagents for this machinery. Thank you for watching. Please post any comments, questions, or suggestions you may have below.